All right. I think we are ready for this first session. My name is Dita De Montis. I'm from Aarhus University, and I'm also a part of the NFF Center. And I'm chairing this session, and I have been really looking forward to hearing these three speakers. And the session is on scaling variant interpretation. And the first speaker is Ruth Loos, and Ruth is professor at the Novo Nordisk Foundation Center for Metabolic Research at the University of Copenhagen and the Novo Nordisk Foundation Center for Genomic Mechanisms of Disease at the Broad Institute. So welcome, Ruth.
Good morning. Thank you so much for having me, and thank you for the introduction, Dita. My talk is on the genetics of obesity, and more specifically in how we actually should curate our phenotype, in this case, obesity, to get to the next level of variant to function translation. So very briefly, my disclosures have no effect on anything I tell you. So no secrets here. Obesity is a traditional common disease, just like type 2 diabetes, cardiovascular disease. The environment has an important impact. Over the past 30, 40 years, the prevalence of obesity has doubled, tripled, and that's definitely driven by a changing environment. We eat more, we move less, but it's in particular people who are genetically susceptible who will respond to this environment and who will gain more than people who are not so much susceptible. So the heritability has been estimated to be 40 to 70%, which is considered, and again, very similar to other complex diseases. Maybe different from other complex diseases is that obesity is a very visible public disease, but I'm not going to talk about that today. But it's also a very early onset disease, unlike type 2 diabetes and cardiovascular disease. So that's when we think about prevention, that's something we need to take into account. Furthermore, it's also a disease that involves a lot of organs or tissues, not just the brain, not just the fat cell, but when it comes to it's like connecting obesity with comorbidities, many other tissues and organs are involved. When it comes to gene discovery, it's been actually a very successful pathway, I would say. It's like we started in 2007 with the discovery of FTO with only a sample size of 5,000 individuals. And then over time, we've identified more and more variants, mostly because we increased the sample size more and more. And many of these efforts have been driven by the giant consortiums, like headed by Joel Hirschhorn. And by now, we have more than 1,700 variants that have been identified, mostly associated with body mass index, or first identified for body mass index. A few have been identified for other adiposity-related phenotypes. And what we've learned in the context of the biology is that the brain is a central organ, probably affecting like our hunger, satiety, reward systems in the brain. The hippocampus, limbic systems are driving this whole process. You see here, this is a nice figure from 2015, but it is still the case. Not much has changed since then. But the variant to function translation has been very slow. FTO was the first discovered phenotype almost 20 years or 18 years now. You see a very nice signal. This is again from an older paper where the p-value is 10 to the minus 60. Now we're at like 10 to the minus 200. And initially we thought that FTO was the causal gene, and there was quite a few papers in Cell, in Nature Genetics that showed very credible results for FTO as a causal gene within the context of obesity. But soon after the discovery, researchers started to venture out. Like some thought it was RP-GRIP1L that was the causal gene because it's like encodes the basal body of the primary cilia, which we know from the Bardet-Biedl syndrome may be cause of obesity. But like others, including Melina's team, thought it was more IRX3 or even further down IRX5 that was the causal gene. So there was a little bit of discussion in the field, but then over time, things got clearer, and maybe this story got concluded with the paper in Science by the Marcelo Nobrego and Melina's team, where they indeed concluded that the signal here in FGO within neurons and pre-adipocytes seem to reach to IRX3 and IRX5, and that may be sort of the causal genes within this locus. And this is just an example to tell you, it's like it took them quite a few, it's like it took them about 14 years to come to that conclusion. And there's 1,700 variants actually that are waiting to be translated. Maybe there's like a dozen of variants that have been translated from variant to function within the context of obesity, but that's about it. Within my team, we've tried to approach the variant to function in a more it's like bioinformatic way, it's like a statistical way, because I don't do actually lab work. So Diane used, I think, about 8 approaches that were at the time that she was doing these analyses to do the gene prioritization. And then she counted how much the same gene was prioritized by the different approaches. So here summarized in this upset plot, for example, here you see that here are the different approaches that she considered for the gene prioritization. And you see that 86 genes were prioritized by 4 approaches, and could be any combination. 34 genes by 5, and 6 genes by 6. So that's not all approaches, but these are these 6 genes, and I won't go into the detail, but some of them are indeed like confirmed, or not necessarily confirmed, but also supported by animal studies that they may play a role in obesity context, and not always, they do not always point to the brain, but often also like adipocyte biology. And then another project that is in process, and that's also like a collaboration with Melina's team, is Virginia. She's doing using a more multi-omics approach, where we go from SNP to BMI through maybe gene expression and proteomics data. So what she did is she combined all the publicly available eQTL data across multiple tissues. Sometimes she did a meta-analysis. She did the same for the proteomics data and used that in her analyses to basically align all the variants that we have. And I should have emphasized, these are the 500 variants that have been reported in the Yang et al. Paper, which is the latest giant paper. She prioritized 413 genes in 189 loci. Again, an upset plot, but the main message here is that, again, the brain remains the most, the tissue in which the most genes are being prioritized or seem to be playing a role. But other tissues are now also popping up. She then overlaid it. So here are her results based on the colocalization with eQTL and pQTL data. She overlaid that with the data from Diane right here, and then overlay that also with the results from CS2G. And you see there's 37 genes that are prioritized by all 3 methods. And now she focuses on these 37. Here are some of them to really summarize the data in a paper that will be coming out soon. So this is a plot that we use where we have a variant, the genes that are prioritized in which tissue. So it's just a lot of data to really summarize into one nice story. But that's coming up, because what's next is actually like the giant consortium has been working. So I haven't updated this plot since 2021, and that's not because I've been lazy, but it's mostly because there haven't been that many new discovery studies. The giant consortium has been working on a meta-analysis. Giant itself combined the data of 2.1 million individuals and then collaborate with 23andMe. So now we have a meta-analysis of 5.6 million people, and this is so now we have really 9,000 loci actually that we have identified, and the Manhattan plot looks like this. It's funny. So the significance threshold is right here, and like you ask, like sometimes it's like too much data, not necessarily helpful. So what can we do with this kind of data? We thought, like, let's start with the easy things. Instead of variant to function, let's just make a new genetic score. And we published that in the summer. It's a rule of lattice effort. So now we made a polygenic score that works actually really well. It's like better than the previously reported genetic scores. You see here the results in the European ancestry. Like, let's focus on the blue dot here. It's like the explained variance is now 17 0.5%. And we see actually it does pretty well in other ancestries, in Central South Asian, East Asian, Middle Eastern, at mixed American, but still not in populations of African ancestry. So that's definitely needs more work, and maybe a starting point of predominantly European data is not the way to go. The prediction of obesity has gotten better. So here are the results in BioMe, Mount Sinai Biobank, Million Veterans Program, UK Biobank for obesity class 1, which is BMI over 30, then class 2 over 35, and class 3 is over 40. And focus on these results here, then you see that the area under the receiver operating curve is getting pretty good. So it's like 0.7 for just your regular obesity. And you have to keep in mind like that obesity is still 50% heritable, 50% or 50% genetic, 50% environment. So we'll never be perfect in predicting regular or common obesity. So 72 is pretty good. And as we go to more extreme obesity, we get better. And maybe that is because more extreme obesity is maybe even more genetic than just your common forms of obesity. And we see this already at a very young age. You see, it's like this is data from the ALSPAC study from 0 to 25 years. And we have 3 curves with the people who are in the lower decile of the polygenic score in red, the people in the top decile in blue, and then the average in yellow. And you see already at age 5 that genetic predisposition is coming through, and it puts you on a track throughout life to be heavier or leaner than the average, depending on where you are. Another thing we're doing right now is led by Victor, who's also here, is just building on the idea that genetics can support drug development. We're using the giant data to look in how many of the loci indeed provide support for for drug discovery. So this is very preliminary data. So out of the 9,000 loci, we prioritize 7,000 genes, and 700 of these genes actually are already the target of either approved drugs or drugs that are close to get onto the market, but not for weight loss. So the question is here, like, how many of these drugs that are approved or in the process of approval could be used for repurposing in the context of weight loss? And then among the new targets that are not currently, or among the new genes that are currently not a target for current drugs, how many could be new targets? And there we use the data from the Illuminating Druggable Genome Initiative. And here we see that again, about 800 genes are predicted to be druggable with high confidence. So maybe among those, there's maybe new anti-obesity targets. Of those 700, so most of them are antibody druggable, and 100 of those are small molecule druggable. Again, very preliminary data, but these are projects basically to avoid the real task, which is the variant to function, right? So because remember, I told you it's maybe a dozen of variants associated with body mass index that have been translated into function, but now we have 9,000. It's a little overwhelming. It's been taking a long time for us to really write up the story into a concise and meaningful paper. And maybe the real problem is not just the amount of data, but is also the phenotype. Some people have tried to shortcut the variant to function by focusing on rare variants. Here you see a few initiatives. The Giant Consortium here used the exome chip quite a while ago. It was a cheap way to get to the rare variants. We had 700,000 individuals find actually 3 variants that are truly rare, below the 1% level, including the 2 variants in the GIP receptor. These are coding variants. A paper by Regeneron that combined data of 600,000 people identified 16 genes, mostly focusing their paper on GPR75. We don't know yet whether that's a real drug target, but they are definitely working on that. And then there's a recent paper by the Cambridge group combining data of 400,000 individuals, and their paper mostly focused on bassoon. This is just to say that this seems an efficient way to go from variant to function within the obesity field, but you still need a lot of data for a few targets. So maybe the problem is not so much with the amount of data. It seems that we have a lot of data, but maybe the problem is really with the phenotype, body mass index. Everyone agrees that it's actually not a very good phenotype because it's pretty crude and it's far from the real biology. So in my group, we've really been focusing on curating the phenotypes, focusing on more refined adiposity traits. One study is focused on body composition, really, and tomorrow, Victor will talk about this. I think it's like an exciting study where we look at variants associated with muscle mass and fat mass in the same direction or in the opposite direction. He'll tell you more about that. I'll tell you a little more about the uncoupling of obesity from its comorbidities, and that's coming next. But we're also focusing on using PET-CT scan data from medical records of Copenhagen Hospital to quantify brown adipose tissue activity, but also other adiposity phenotypes. And that's very much in development as well. So we use AI to do the quantification. And we're also focusing on fat distribution. So obesity it's not only about too much adiposity, but also where exactly you will store it. And we also increasingly focusing on biomarkers, particularly with the proteomics becoming increasingly available. This promises to be an exciting field as well. So today I want to focus on this story, and actually it just got published last week in Nature Medicine, and is led by Nathalie Chamy, who's here as well today, and by J.J. Wang. So what we did is we did not do a case control analysis of healthy obese individuals versus unhealthy obese individuals, because there's a lot of discussion about that. But we looked at these phenotypes, body mass index, body fat percentage, and waist tip ratio on one hand, and these cardiometabolic risk factors on the other hand. So lipids, blood pressure, and glucose and insulin. And what we did is, so this story actually builds on a previous story where we use summary statistics. So here we use individual level data of mostly of the UK Biobank. So what we did is we take a distribution of body mass index. It's right skewed, but we made a standard normal distribution with a mean of 0, standard deviation of 1. And we did the same for all the traits. And also for the cardiometabolic traits. So each phenotype is now on the same distribution. And we made a new phenotype by subtracting one from the other. Say my body mass index is +3 on the BMI side, but -3 on the LDL side. So if you subtract one from the other, depending on which one you subtract, now I'm all of a sudden +6. So that becomes a whole new distribution. And now we did a GWAS on that new distribution. So if I'm +6, it means that I'm a healthy obese. I'm a person with healthy obesity because I have a high BMI and low LDL. If you're -6, you're the opposite. You may have a low body mass index, but you have a high LDL. And we did that for BMI and LDL, but we did that for all these possible combinations. So we created 24 new phenotypes. We did a GWAS across these 24 new phenotypes. We made sure that the signals we observed are co-localized, and we ended up with 205 new loci in 206, with 206 independent association signals. So just to confirm, we then made a score of these 206 loci, or 266 loci. So we have the uncoupling score, because we really look at how adiposity is uncoupled from these cardiometabolic risk factors. And we compared it with your typical unhealthy obesity score, you could say it. So red is the healthy score, blue is the unhealthy score. And you indeed see, like, if you do the association within the UK Biobank, there's no effect on height, but you quickly see that the difference is particularly visible in the VAT-SAT ratio, where if you have an unhealthy score, there's not so much an effect, and here's the null line. But if you have a healthy score, you actually have a favorable fat distribution. And you see the same for waist to hip ratio, where the unhealthy score is associated with a higher waist hip ratio, where the healthy score does not have too much an effect on fat distribution. It's more visible if you look at the risk factors, where here is the null line, and you see that the healthy score lies mostly on the inside on the negative side, so the protective side of this radar plot, meaning it's associated with lower blood pressure, lower glucose levels, lower HbA1c triglyceride, and of course higher HDL as expected. Whereas the unhealthy score is also as expected, lies on the outside. So this is just for confirming that what we find is indeed healthy obesity versus unhealthy obesity. We did a PheWAS within UK Biobank, and indeed we see like a nice confirmation that both scores are associated with increased obesity, but the unhealthy score is indeed associated with higher risk of type 2 diabetes, hypertension, and the healthy score has a protective effect. Of interest is, of course, that the association with other diseases, not the non-cardiometabolic diseases, that seem to be associated with weight-bearing diseases such as cellulitis, varicose veins, and so much. So it's of course a bit tricky to call it healthy and unhealthy, because in the end, the risks of the people with a high healthy obesity score, they're still associated with these diseases in the same way as the unhealthy score. We also confirmed these results in children, focus on the cardiometabolic risk factors. If children with a healthy score, and they're at around the age of 11 and 12, they already at that age, you see that they have a protective profile compared to children with a high score on the unhealthy score. If you look at the cardiometabolic, or if you look at tissue enrichment analyses with a healthy variance, it's particularly the peripheral tissues that show up, adipose tissue, abdominal fat, and so on, but also cartilage, adrenal, stomach, whereas the unhealthy score, similar to the BMI score, is associated with the central nervous system. Right? So clearly different biology that is being observed. We also look at the proteins. We know from the UK Biobank data that high body mass index is associated with high protein levels, probably because the bigger you are, the higher the levels. That makes sense, and that's shown in this data from Sun et al. So when we looked at the scores and their association with the UK Biobank proteins, the unhealthy score, which has some more power, is associated with more proteins. The unhealthy score is also associated with 337 proteins. 208 are associated are overlapping. Of the overlapping proteins, 85% are in the same direction. And this is what you see here. This is somewhat like there's 3 dimensions to this plot. On the right-hand side means higher adiposity, higher cardiometabolic disease for the red dots. But for the yellow dots, it means like a higher adiposity, lower cardiometabolic risk. And on the left-hand side, lower adiposity, lower cardiometabolic risk for the red dots again. And for the yellow dots, lower adiposity, higher cardiometabolic risk. So these 176 proteins are directionally consistent with the traditional adiposity proteins. Leptin, fatty acid binding proteins, you see them pop up in at both plots at the same time, and the same like with some others on this side. So we call these proteins are adiposity driven. What about the 15% others? There's about 32 proteins that are actually going in the opposite direction. So they will show up here in the red dots, but here on the yellow dots, and on this side on the red dots, and here on the yellow dots. So, for example, growth hormone receptor, LDL receptor, ApoF, FGF21. Too much data to really go into the detail right now, but for us, it suggests that they're not driven by adiposity as such, because then they would lie on the same side, but now they lie on the other side. And what could explain here is that they're driven by the health status, the cardiometabolic health status. And then there's another, and then go a little faster, there's 129 proteins that only show up for the uncoupling, even though this uncoupling score has less power than the unhealthy score. We see AdipoQ as expected to show up, lipoprotein lipase, myostatin. So there's a lot of interesting proteins that are showing up to be associated with either score, telling us more about the biology. Going a little faster, I showed you that we have one set of 206 genes that are associated, or variants that are associated with this healthy profile, but actually they also cluster within multiple subclusters. For example, here, it's a heat plot. These are the 266 variants associated body fat percentage and body mass index. And by definition, because that's how we discovered them, with a protective profile with any of these cardiometabolic risk factors. But you see they clusters. This cluster 3 is associated with lower body fat, lower blood pressure. Cluster 5 with lower glycemic traits. Cluster 6 with a healthier lipid profile. Cluster 1 is pretty complicated because it has a healthy profile for triglycerides, but unhealthy for glycemic traits. And then 4, 7, and 8, they're actually more globally associated with a healthier profile. So combining, and then we did a variant to function prioritization, bioinformatically overlaid with GTEx data, and then in collaboration with Melina, also using CS2G. And combining all that data and knowing the clusters in which these variants fall, and sort of having that more refined phenotype for each of these variants, we were able to prioritize some of these genes better than we could without that information. So here I'm just going through it, highlighting some of these genes. If any of these genes are familiar, like, please let me know, because we are now working on like the real variant to function. So even though this study was published, the study is not complete, and we're working with Melina's team, and one posted in my group, Melina, Eleonora Manita is here doing this real variant to function in the lab with Melina's team. This is just to say, and maybe to conclude, that it's like gene discovery is maybe complete, but we really need to work more on curating the phenotype to get the phenotypes closer to the biology, to facilitate and make it much easier to do the variant to function translation. So it's important to just not forget about the phenotype. So just to point out that the biology that we implicated with the genes that we have identified are the ones that we've identified before. But actually now with the more data, we strengthened these observations, and we actually identified a few new pathways that seem important in the variant to function translation. Just to say, like, identifying variants is not hard, but genetics may help elucidate the heterogeneity within obesity or any complex trait. We see multiple loci that uncouple adiposity from its comorbidities. Multiple mechanisms seem to be underlying, and by combining this better description of the phenotype with the variant to function translation, I think we may expedite the actual variant to biology observations. Very grateful to my team at CBMR, at the ICON Institute as well. Working with Melina has been a blessing, and it really has pushed me to the next level of the variant to function translation and going beyond the bioinformatics. And of course, also the giant consortium with Joel Hirschhorn leading it, and of course, the funding for this work. I'll finish here.
Thank you, Ruth, for a very, very interesting presentation. So are there any questions from the audience?
So a couple of questions, Ruth. So one, if you were to filter genes by just the number of genes that definitively have multiple variants that can be linked to the phenotypes of interest, does that help narrow the list? So I'll ask 3 quick questions. So that's question number 1. Question number 2 is, I never saw GLP-1 come up on any of your gene lists. So if you were to do a retrospective on GLP-1, where would it have fallen out kind of in this list? And then the final question is on FGF21, it looks like it goes in one direction and then the other direction. So it increases risk of adiposity and then decreases risk of cardiometabolic disease or cardiovascular disease. So if you were to perturb that, would you expect differential effects across those 2 phenotypes?
Right. Actually, I did the first question about filtering genes. What was exactly what you mean?
Multiple alleles at a locus to help whittle those down to small?
That's a good question. Actually, we haven't looked into that. But indeed, we are working on the variant of function in the lab with Melina. Don't know what the stage is at the moment, but yeah, waiting for the data. For GLP-1 receptor variants, it's very interesting with the 9 million, with the 5 million meta-analyses, it's starting to show up. It's a very nice peak on GLP-1 receptors, but it's the first time we see it in a meta-analysis show up. In adults, it's very intriguing, but a small 28,000 individuals meta-analyses in children, but in children before the age of 3, GLP-1 receptor variants are showing up there. And that's actually a phenotype. BMI before the age of 3 is not correlated with your adult phenotype. So it's very intriguing. We don't quite know what's happening. But then we need 5 million people to find it in adults. And then your third question, I already forgot. Yeah, it's true. It's like, and I didn't mention it here, but these are plasma proteins, and these are still very correlated phenotypes or correlated observations. We haven't looked really at the causality. Is FGF21 up as a causal or as a consequence or as a compensatory? It's like there's a few other of these proteins like LPL and myostatin that are in the direction that we didn't really hypothesize would be, but that maybe have to do with the fact that it's plasma proteins.